lang. Wait lang. Screen na lang siguro. Cancel. Okay, again, good evening to everyone. So, for this session, we're going to discuss about lesson 3 na, 'di ba? Wait lang. Ay, na-wrong ko. Stop share, then share, tab. Midterm. Ako naman na rin. Ha? Well, anyway, share screen ko lang muna, ha? Window. Sa window siguro na lang. Actually. Okay, so again, we are now on lesson 3. So, for this session, we're just going to discuss about photosynthesis, no, sa lesson 3. And I will have a video presentation on photosynthesis at respiration for lesson 3. And for lesson 3, we're going to discuss on photosynthesis and cellular respiration. No, the importance and the factors that are necessary for the photosynthesis, synthetic and cellular respiration. At saka, differentiate these crops based on their photosynthetic and cellular respiration. So, in physiology, we have two processes, physiological processes that are very important. Now we have photosynthesis and respiration. And this is involved in the manufacture of food and the utilization for its growth and development. So to learn actually more about photosynthesis, I have a more interactive way in presenting them in one of my favorite videos. But then you can always go back to this module for references, no? So at the end, you're going to answer this question. Now, what is the importance of photosynthesis in horticultural crops and to animals? And why cellular respiration happens? And how photosynthesis and cellular respiration differ from each other? And we shall have next lesson by next meeting. No? So I guess we will start now. Screenshots ako sa mga okay. So this is one of my favorite na video. Ano lang siya?
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about photosynthesis. I love photosynthesis because it gives me two things that I need. I need to breathe, so it gives me oxygen, and I need to eat, and so it's going to give me food. And so I love photosynthesis. You might think it's only found in these things, plants, but it's also found in bacteria, it's found in algae, and so it's found in protists, it's found everywhere. And so photosynthesis has been around a long time. It's super important that you understand how it works. And so let's start with the site in eukaryotic cells of photosynthesis, and that's the chloroplast. So this is a number of cells, and you can see how many chloroplasts we could have in a typical cell. So there's a whole bunch of them. There are a few terms that you should be familiar with and where they are. First one is a thylakoid membrane. Thylakoid membrane is going to be organized like this. And basically, that's where the light reaction is going to take place. If you got a stack of thylakoids like this together, we call that a grana. The other big thing to understand photosynthesis is that this is filled with a liquid, and that liquid is called the stroma. That's going to be the site of the Calvin cycle. If we were to grind up a leaf, what we would find is that there's not only one pigment, chlorophyll A, that does photosynthesis, but there's a number of them that are working together. And so if you grind up a leaf into some chromatography paper, and then you put it in a solvent, what you'll get is chromatography. It's going to separate into all its different parts. And so this right here would be chlorophyll A and chlorophyll B, and this would be like carotene and xanthophylls, and they're all working together. You'll see these other pigments in the fall when the chlorophyll moves back into the leaf and is reabsorbed. But if we look at what light they absorb, here's chlorophyll A and here's B. This is what's called their absorption spectrum, what color of light they're able to absorb. And you can see that they absorb a lot of the blue, a lot of the red, but they don't absorb a lot of this in the middle, this green. And so a quick question could be, what is their least favorite color, plants? And the right answer would be green, because they reflect that green light. Now, this has actually puzzled scientists for a long time, and we really don't have a definitive answer as to why plants are green. Know this, that if they were black, they probably would get a little bit too hot. They would absorb too much light. And so let's start with an equation, because this is simply a chemical reaction. It's a chemical reaction with a number of steps. But what are the reactants? Water and carbon dioxide. And so how does a plant grow? It's basically taking water in from its roots, and it's taking carbon dioxide in through its leaves, through its stomata. The other thing it needs is light. And so it's just taking these simple ingredients, and then it's weaving those together into glucose, this monster molecule here, and then oxygen. And so this is the food that I get, and this is the oxygen that I breathe. Now, are plants just nice? No, they're making this sugar for themselves, so they can break it down using cellular respiration. And in fact, if I put this arrow in the other direction, that becomes cellular respiration. So they're making food for themselves, and they're also going to make some of the structures. So like the cellulose in the cell walls of a plant is made from that as well. Okay, so whenever I try to think what are the different steps in photosynthesis, I always imagine this picture right here. There's photo and synthesis in the word. Photo means light, and synthesis means to make. And so there are two steps in photosynthesis. The light reaction, and those are going to take place in the thylakoid membrane, and then the Calvin cycle. We used to call this the dark reactions, which is a silly term. Doesn't happen during the dark, it happens during the light. And so basically, the person who worked this all out is Melvin Calvin, and so we named it after him. Whereas this takes place, you guessed it, takes place in the stroma, or this liquid portion. And so let's kind of do a cartoon version of photosynthesis. What are the reactants again? Water, light, and carbon dioxide. What are going to be the products that come out of this? It's going to be oxygen and glucose. So let's watch what happens. In the light-dependent reaction, water and light go into the thylakoid membrane, and they produce two things. They produce oxygen. Oxygen is simply a waste product. And then they're going to produce these chemicals, NADPH and ATP. So they have energy now. Let's watch what happens to them. Well, the energy is going to transfer to the Calvin cycle, where carbon dioxide comes in and then glucose goes out. And so this is the big picture of photosynthesis. But now let's kind of dig in a little bit deeper and talk about the light reaction. Okay, so where are we? We're in the thylakoid membrane, so we're in this membrane right here. So if we were to zoom in to that membrane right here, that's what this diagram is. Okay, so what are the two things coming in? Well, the first one's going to be light. So light's coming in here, light's coming in here. What's the next thing that we're going to have coming in? And that's going to be water. Okay, so let's look at some of the other big features in this thylakoid membrane. So this is the outside, or the stroma, and this is going to be the lumen, or the inside. And so there's a couple of big things right here. What's in here? Well, these are basically going to be proteins with chlorophyll on the inside of it. And so we call that whole thing together a photosystem. So this first one is actually called photosystem 2, and then we go to photosystem 1. And the reason we go backwards is that photosystem 1 was discovered first. So basically, what comes in? Light. What's that light used to do? Well, that light is used to power the movement of an electron through an electron transport chain. So that electron is going through proteins, carrier proteins, and eventually that electron is going to go to here. It's going to go to NADPH. Because remember, that's one of the products of the light-dependent reaction. Okay, what happens to the water then? So the water is going to be split right away. If you split water, what do you get? Well, you get oxygen. So that's the O2 that's going to diffuse out of a cell, and that's the oxygen that you're actually breathing right now. And then we're going to have these protons, which are simply hydrogen ions. So they're hydrogen atoms that have lost their electron. Okay, so this is getting kind of messy. So let's look what happens next. As that electron moves through the electron transport chain, and again, it's powered by the introduction of light here and light here, that electron is going to be moving all the way down here, and every time it goes through one of these proteins, it's pumping protons to the inside. So it's pumping protons to the inside. Now, protons have a positive charge. So basically what's happening is that you're building up a positive charge on the inside. So there's a positive charge in here. If you know how cellular respiration works, you'll realize that this is the opposite of that. So now we have all these positive charges on the inside. Where do they go? Well, there's only one hole that they can go through, and that is to go through this protein here. As those protons move out, they're moving through a protein called ATP synthase, and it works almost like a little rotor. And every time a proton goes through, we make another ATP. So what have we made in the light-dependent reaction? We've made NADPH and we've made ATP. And what's nice about that is they're now just sitting right here in the stroma, and so they're able to go on to the Calvin cycle, which is going to be the next step in this process. And so who's providing the energy? Light. Who's providing the electrons? Water. And then a base or a waste product of that is simply going to be oxygen. Okay, let's go to the Calvin cycle then. So what's happening in the Calvin cycle? You can see here's those reactants. So we've got our ATP here, and NADPH. What are they providing? Simply energy. We also have this molecule here. It's called RUBP. Basically, it's a 5-carbon molecule. And then we have carbon dioxide coming in. So it moves through the stomata of the leaf and it's going to diffuse its way in. Carbon dioxide is a 1-carbon molecule. So basically there's an enzyme here called Rubisco, and it's going to attach this 1-carbon molecule to a 5-carbon molecule. It immediately breaks into 2 3-carbon molecules, and then it gets energy from ATP and NADPH. And when we're done, it's creating this chemical down here called G3P. What does G3P become? Well, it can be assembled quickly into glucose or sucrose or maltose or whatever they need to do. That's going to be produced right in here by the G3P. So that's where we're synthesizing. In other words, we're taking carbon and we're fixing it. We're making it usable. Now, some of that G3P is released, but a lot of it is recycled again to make more of this RUBP. And so that's why it's a cycle over and over again. What's the big picture? If we don't have ATP, if we don't have NADPH, then this process is going to shut down. What's the other thing that could shut it down if we don't have carbon dioxide? Okay, so that's basically photosynthesis. And again, it's been working for billions of years. But there's a slight problem, and that problem is called photorespiration. What is photorespiration? Well, photorespiration occurs only when we don't have enough carbon dioxide. So if we don't have enough carbon dioxide, let me cross that out. Well, we certainly can't make our G3P, but something worse happens. Oxygen can actually jump into the Calvin cycle and using Rubisco can form another chemical. Now that chemical doesn't do anything. In other words, it has no purpose and the cell actually has to break it down. And so as a result of that, plants, and we call almost all plants C3 plants, and the reason we call them C3 plants is this G3P is going to be a 3-carbon molecule. So these C3 plants, photorespiration is bad. In other words, they don't get anything out of it. And so they're going to lose based on that oxygen kind of jumping into the Calvin cycle. And so you might think evolutionarily, why would this have even evolved? Well, remember, photosynthesis shows up first, and then oxygen in the atmosphere shows up much later. And so it wasn't a problem initially, but it became a problem. Another question you might think is, well, when are we not going to have enough carbon dioxide? When wouldn't we have carbon dioxide? Well, how do they get carbon dioxide? A plant is going to have a stomata, and it's surrounded by guard cells. And so basically, when a plant opens up its stomata, carbon dioxide can diffuse in. And so the only time the plant wouldn't have carbon dioxide, because we have tons of carbon dioxide in the atmosphere, is when it's actually closed. And when would it be closed in a plant? The only time it's closed is when it's really, really hot, and a plant doesn't want to lose water, because through transpiration, you're constantly losing water. And so if you're a plant, you have this, if it's a hot day, you have this really tough choice. If you open up your stomata, you're going to lose water, you could shrivel up. If you close it, you can't get carbon dioxide in, and then you're going to start doing photorespiration. And so, of course, nature has come up with solutions to this over time. And it's only going to be found in plants that live in a really hot environment. So here's the first solution. And this totally makes sense. So this is in camp plants. Camp plants, an example would be a jade plant or like a pineapple. Basically what they do is they only open their stomata at night. And so at night they open up their stomata, and then the carbon dioxide will come in and they'll create malic acid out of it. So they're going to store it in vacuoles inside the cell. Okay, so now when it's daytime, what they can do is they can close the stomata because they don't want to lose water. And now they can actually take that carbon dioxide out of the malic acid, and they can use it in the Calvin cycle to make sugars. So the great thing about a camp plant is, again, they're only taking in carbon dioxide at night when it's cool, and then during the day they can close their stomata and they don't lose water. Another example of this would be in C4 plants. What they do is, instead of doing it day and night, what they'll do is they'll take that carbon dioxide in, and they'll actually use enzymes to make a 4-carbon molecule out of it. That 4-carbon molecule will move to some cells on the inside of the leaf called the bundle sheath cells, and then they can simply introduce carbon dioxide into the Calvin cycle here. And so again, both of these solutions are basically taking in carbon dioxide when you can get it, creating a chemical out of it, and then they can introduce that chemical into the Calvin cycle, and they don't have to wait for carbon dioxide to diffuse in. Now, of course, there's going to be extra steps in here, so it's going to require more energy, and so we only see this in areas where it's really, really warm. But an example of a C4 plant that we all eat and use a lot of, in fact, most of us are just made out of this stuff, is corn. And so that's photosynthesis. A simple problem is photorespiration, but I hope that's helpful.
See what your love and total expert protection can help do. New Nido 3 Plus.
Okay, so before we proceed with um, cellular respiration, so I just have to highlight some important points on photosynthesis. So we have actually, first question is that does photosynthesis exist in with light, or I mean without light? Does photosynthesis occur without light? Anyone? Mag-occur ba ang photosynthesis kahit walang light? Yes or no? Anyone would like to raise your hand? Or yes, Sir Adrian. I don't know. By the way, hello ma'am. Hinay ang volume, ma'am, ha? Really? Hinay akong volume. Full naman akong volume ba? Hinay gapon. Hello?
Kusog-kusog man, ma'am.
Okay. So, unsa man, Ludi, yes or no? Mag-occur ba ang photosynthesis without light? Yes or no? Ay, murag si Adrian ang ang tawag na. Walay signal. So, Ludi or anyone, yes or no? Of course, yes, no? Doon sa ating reviews, photosynthesis has two phases, no, the light reaction and the dark reaction. And in the light reaction, it is when through the help ng light or yung sunlight, no, that na yung photosynthesis will start, no, through the light, through the collection in the stomata, no, I mean thylakoid or the chlorophyll. And during the dark reaction naman, it could be without light, pero yung sa Krebs cycle na, 'di ba? And it was mentioned how the Krebs cycle and the output of Krebs cycle is a Krebs cycle, I mean Calvin cycle, sorry, um, is what we call yung 3 glyphosate, no? 3. That's why most of our plants are 3 plants kasi 3 carbon siya na glyphosate. And for that, that's the common photosynthesis. So anong kaibahan? And so there are plants that merong mga specialized structure. So we have the C3, C4, and CAM plants. So anong kaibahan ng tatlo? Anyone? Based on sa ating nakitang video. Yes, Roy. By the way, this video, this call is being recorded and I can access. I will send you the recordings through YouTube at saka meron din tayong kasi I'm using the Google Meet, so we can access your attendance with specific details kung kailan kayo nag-in and out and so on. So mas better siya in recording. So again, we have C3, C4, and CAM plants. So anong kaibahan ng tatlo? Alin doon sa different 3 types ang pinaka-efficient in terms of photosynthesis? Yes, Steve. How about Catherine Delgado? Okay, mahina siguro ang signal natin kasi it's raining, 'di ba? So anyway, so yung most better or more efficient than C3 kasi C3 yung mga normal like the usual plants that we have, no? Are the C4 and the CAM plants. So C, for example, yung corn, 'di ba? Kasi meron silang, what do they have? Specialized anatomy, no? Leaf anatomy. And they have that crans anatomy. Specifically, meron silang bundle sheet cells which make use in producing the C4, the carbon 4 product ng Calvin cycle. While yung CAM plants naman are those mga cactus or mga plants that thrives in the desert kasi they don't have the crans anatomy, they don't have the bundle sheet, but they have the survival or the ability that they're going to close their stomata during night. I mean, during day, and they will open the stomata during night para doon na sila mag-take in ng carbon dioxide which is needed in photosynthesis. So, that's why mas efficient sila, mas madami silang ATP na napoproduce. So, yun lang, no, yung mga highlights of photosynthesis. And, the 3 types based on photosynthesis, we have the C3, C4, and CAM plants. And, if you have any questions or clarification before we proceed with cellular respiration. So if none, then let's watch this. And don't forget na Do you have assignment no after lesson 3?
Hi, it's Mr. Anderson. In this podcast, I'm going to talk about cellular respiration. Sometimes students confuse respiration, breathing, just breathing in, and cellular respiration, and they are linked. Cellular respiration, however, is going to take place at the level of the cell, more specifically inside the mitochondria. And so we need oxygen for that. But it's basically taking our food and then breaking it down in the presence of oxygen to make ATP out of it. What if you're a bacteria? Can you do respiration? You sure can. You don't need a mitochondria to do respiration. They can actually use their outer membranes to do aerobic respiration. And so basically, if you are a track athlete, so this is Usain Bolt right here, when you run, you're using respiration to make energy in the form of ATP and allow your muscles to move. And so this is a quick little study I did. I took all of the world records currently right now from the 100 to the 10,000. And so this is Bekele is the guy who owns the 10,000 meter record. And what I did is I figured out their pace. In other words, how fast they're running. This would be in meters per second for the 100, the 200, all the way down to the 10,000 meter run, which is a little over 6 miles. And what you can see in this graph is that the pace is quickly going to drop off, and then it's pretty much going to stabilize. And if we were to go out to the marathon or continue, basically we're pretty effective at running at a specific pace, but we die really quickly when we're sprinting. And so a way to think about that is the two things we're going to talk about in this podcast is we're going to talk about aerobic respiration. So aerobic respiration is going to be respiration in the presence of oxygen, but we also have almost like a turbo button that is anaerobic respiration. And so if we really need to go fast, we can get that extra speed that we have up here by doing our anaerobic respiration. The problem, if you've ever run, is that when you get out to this 400 meter, you get a buildup of lactic acid, and it's incredibly painful, and so you can't keep that pace going. Example of a lab I did in class was, and it's weird how this exactly mirrors it, what we did was the muscle fatigue lab. So basically you had a tennis ball, and in one hand you had to squeeze it as many times as you could in 10 seconds, and then do another 10 seconds, and do another 10 seconds, and this is the class average. So the class average was, it looks like around 25 times in 10 seconds. But you can see that it quickly drops off, and then it kind of levels off. And so the same thing, this would be that aerobic respiration, and then this is going to be that anaerobic respiration. And it was fun to see the students' faces, because as they start to go anaerobic, your arm just starts to build up if you have lactic acid on the inside of it. But before we get there, let's talk about respiration and what it's for. It's for heterotrophs. So we're heterotrophs, and basically what we're doing is we're taking organic compounds in the presence of oxygen, and we're converting that to carbon dioxide and water. What else are we generating? ATP. Now, what kind of things are doing this? Animals, fungi, bacteria are all heterotrophs, and they're using the organic material to actually make energy. Luckily, we have autotrophs like plants and algae, and basically what they're doing is they're converting that carbon dioxide and water back into organic materials. The only thing that's a little deceptive is that plants are also going to break down those organic compounds, and so they do cellular respiration as well. And so everything's doing cellular respiration. It's how we get energy out of our food. Okay, here's our equation. And again, if you know what photosynthesis is, this is simply the opposite. We're going to take glucose in the presence of oxygen. So here's glucose and here's O2. And then we're going to break that into carbon dioxide, water, and then we're going to generate a little bit of ATP. Now, where is the energy sit? The energy sits right here in this hydrogen on the outside of that glucose. And watch what happens to that hydrogen. It's going to fall down and it's going to grab onto the oxygen, because oxygen wants electrons. And so that's where the energy is coming from. And what the energy is used to do is it's used to make ATP. And ATP is that little fuel that we use in all of our cells. This slide is funny, but it's saying this: behold the power of oxygen. So this fire comes from oxygen pulling electrons close to it. And so there's a huge amount of energy found inside that pull of electrons towards oxygen. Now, if we were to do this inside our body, we could get a lot of energy out of our food, but we would also burst into flames. And so we do it in a really controlled process. Just like when we learned photosynthesis and we had to learn the parts of the chloroplast, when you're learning the cellular respiration, you have to learn a few parts of the mitochondria. So first of all, we have these folds on the inside of the mitochondria. Those are called the Cristae. And basically what we have is two membranes. We're going to have an inner membrane right here, and then we're going to have an outer membrane right here. And then this space in the middle is called the inner membrane space. And on the inside, mitochondria, we think, used to be bacteria of their own. And so they'll reproduce through binary fission. They have their own DNA. They have their ribosomes. But they are kind of almost living inside us, not as a parasite, but as a symbiont. They're actually helping us as we generate energy. So there are 3 steps in cellular respiration. Let's start with the first one. So the first one is going to be glycolysis. The second one normally we think of as the Krebs cycle, and then the third one is going to be the electron transport chain. And so the first one, I love this diagram here because it's putting glycolysis outside the mitochondria. And so this is going to take place outside the mitochondria. Where would that be? Well, that would be in the cytoplasm of a cell inside your body, or it would be right outside of bacteria. But what happens in glycolysis? Basically, we're taking glucose. Glucose is a 6-carbon molecule. And in glycolysis, we're going to break that down into 2 molecules of pyruvate. Each of those have 3 carbons inside it. So the 2 3-carbon molecule, that's what glycolysis does. What do we generate in there? Well, we generate a little bit of ATP. For 1 glucose molecule in glycolysis, we're going to make 2 ATP. The other thing that we make is a chemical called NAD. What we're basically doing is we're transferring high-energy electrons to NAD, and we're adding protons to it as well. And we'll get to NAD in just a little bit. Let's follow pyruvate then. Pyruvate is going to diffuse into the mitochondria, and then we're going to have this pyruvate dehydrogenase complex. And basically what it's going to do is it's going to convert that 3-carbon molecule into acetyl-CoA. This is coenzyme A. So basically now we have a 2-carbon molecule. That is going to go into the Krebs cycle. Now, since we're going from a 3-carbon pyruvate to a 2-carbon acetyl-CoA, we're giving off carbon. And that carbon is going to be given off in the form of carbon dioxide. And so when you breathe out, a third of that carbon coming out of you is going to come right here from this complex inside the matrix, we call this. I should have said that before. This is the matrix on the inside of the mitochondria. Okay, let's keep watching acetyl-CoA. So it's a 2-carbon molecule. Where does it go next? It goes to the Krebs cycle. And so in the Krebs cycle, we're going to break it down further, and we're going to get rid of these 2 carbons in acetyl-CoA, and we're going to give those off as carbon dioxide. So we're getting rid of carbon dioxide. What else are we producing in the Krebs cycle? You can see here that we're producing a little bit of ATP, 2 ATP, but we're also adding energy, again, to NAD. And we're adding energy to its friend, we'll call this FAD. And so what do NAD and FAD have? They have these high energy electrons, and they're going to carry those electrons to the 3rd step, which is the electron transport chain. Okay, let's get to the electron transport chain then. All of our energy pretty much that was in glucose is now in NAD and FAD. So they're going to transfer their electrons, and those electrons are going to go through what's called an electron transport chain. Basically, they're moving through a series of proteins, and the energy of those proteins is used to pump protons. Protons are going to be hydrogen ions to the outside of this inner membrane, into what's called the inner membrane space. So now we've built up all of these protons right here. What happens to the electron? The electron is going to be added to other protons and oxygen that we breathe in, and that's going to make our byproduct, which is going to be water. And so let's slow that for just a second. The oxygen that you breathe is moving in here, and it's going to be that last electron acceptor right in here in the matrix, and we're going to take the protons. What happened to those protons? They'll actually flow through a protein called ATP synthase. Those protons will combine with the electrons and the oxygen, and it's going to make water, which is going to be a byproduct of that. Now, how much ATP do we make down here? Well, we can make around 32 or 34 ATP in this last step. And so in the electron transport chain, we're making a whole heck of a lot of energy. And so it's worth taking a look at how that actually works. So let's go to the electron transport chain. So kind of to situate ourselves, what do we have? Well, we have NAD, our friends NAD and FAD. What are NAD and FAD passing off? They're electrons. Those electrons are going to move through the electron transport chain like this. Every time they go through one of these proteins, it's going to pump another proton ion out, because that's the other thing that NAD and FAD are bringing. They're bringing these hydrogen ions. So we're going to pump these ions out here, and pretty soon what you get is a heck of a lot of positive charge out here in this inner membrane space. There's no place for it to go. In other words, every NAD that we drop off, we're going to move these electrons down and we're going to generate a whole heck of a lot of positive charge in this intermembrane space. Now, if you look right here, fat is actually dropping its electron a little bit farther down, so it can't generate as much, but we're pushing out either 3 protons or 2 protons, depending on if it's NAD or FAD. Okay, what happens to all of these protons out here? They can't go anywhere. They can't go outside the mitochondria, they can't come inside the matrix, but they can move through this. This is called ATP synthase. That's the name of this protein right here. And basically, this is the site of ATP synthesis. And so basically, as every proton flows through it, every proton that comes through, we're going to generate ATP. And it almost works like a little rotor that every time a proton goes through, it switches it and it attaches that phosphate onto ADP to make ATP. And that's why in the electron transport chain, we can make all of that ATP. There's nothing special about it. It's just that we're storing all that energy, and instead of releasing it in a ball of fire, we're releasing it in little bits to make a heck of a lot of HTP or ATP. Okay, so there's a problem. What happens if you don't have oxygen pulling that electron the whole way, or let's say you don't have mitochondria present? Well, then you have a problem, and the problem is this. It's okay to take glucose during glycolysis and break it into two pyruvates because you're going to make a little bit of ATP, but the problem is that you're adding those electrons to NAD, and so basically what's happening is that we're adding electrons to NAD plus, and we're transferring it to NADH, and so pretty much what happens is there's no more of this. And so glycolysis has to shut down. Even though we can make a little bit of ATP with each breakdown of glucose, eventually there's no NADP plus, and so the whole process has to stop. And so nature, of course, has a solution to this, and the first one is called lactic acid fermentation. This takes place in your muscles, especially when your muscles are under a huge amount of stress, like if you're sprinting or if you're holding your breath for a long period of time. And so basically what's going on, again, there's no oxygen, there's no mitochondria, so let's look what happens. Basically, your cells are taking glucose in glycolysis and breaking it down into 2 pyruvate molecules. So we were stuck here, remember, with the NADH. But then there's a further conversion. Basically what you're doing is you're converting that pyruvate down into lactate or lactic acid. The nice thing about that is it's accepting these electrons, so we can make more of this NAD plus, and then this can be recycled again. And so basically what happens is that you can have this process occurring with glucose, over and over and over and over and over again. And every time you do that over and over and over and over again, basically you're making 2 ATP each time. And so if you've ever done sprinting, when you're sprinting, you're getting aerobic respiration, but you're also doing anaerobic respiration on top of that. The problem with that is you're going to build up lactate in your muscles. And that lactate is like a toxin. You're going to have to break it down. And that takes oxygen. And so if you've ever watched a sprinter, especially somebody who's run like the 400-meter dash, when they're done and they're interviewing them, they have a hard time doing an interview because they have to keep breathing to take in more O2 and eventually get rid of that lactate. And so lactic acid fermentation is going to take place in some bacteria and in muscle cells, but we have another solution to this in bacteria, anaerobic problem of stopping right here with this full NADH, and that's called alcoholic fermentation. Alcoholic fermentation works the same way. Basically, we break it down into pyruvate, and then we break that further down into a chemical called ethyl alcohol or ethanol. It's accepting these electrons so it can recycle this NADP plus again. The only difference here is that when we made lactate, that was a 3-carbon molecule. When we do alcoholic fermentation, what we're making is carbon dioxide, and we're giving that off. And so if you were to take yeast and put them in a bottle with a bunch of fruit juice, basically what they'll do is they'll use up all the oxygen, then they'll switch to alcoholic fermentation. What are they going to build up? They're going to build up ethyl alcohol. That's simply how we make wine. And there's also going to build up carbon dioxide, which we could let go. Or if we're making beer, that's the carbonation that we're going to find in beer. And so again, cellular respiration is just a quick way to get energy out of glucose. We use glucose as an example, but we can do cellular respiration on pretty much any type of food. And it's a way that we get energy. And we're doing it, bacteria are doing it, plants are doing it. And I hope that's helpful.
Okay, so that's it, no? again, I will share to you your module, no, for lesson 3. So again, talking about respiration, we have learned that after photosynthesis, we have to make use of this um, glucose, no, or sugar, sugar photosynthase to make use of our photosynthase or para for to produce ATP or energy for our growth and development. But before we proceed, um, Sir Roy, you have a question? You're raising your hand. Wala, na wrong lang siguro siya. So going back here, no, as I have shown to you, ayan, the process of photosynthesis and the respiration. Ayan. So overall, photosynthesis, these are the processes in comparison sa respiration. So it's just about producing, from sucrose or glucose through glycolysis, we produce ATP, no, in the mitochondria through respiration. Now, the video will show us and help us understand more why and how does respiration occur in all living things, including humans, plants, animals, bacteria, and fungi. No, and also, after that, we have known that photosynthesis, respiration, occur with or without air, no? So ang usual na respiration ay aerobic respiration. And without oxygen, of course, it becomes anaerobic respiration. And in anaerobic respiration, that's how fermentation is produced, no? And we have a lot of fermented products, diba? And that's the product of anaerobic fermentation. Like for example, no, kung if ang product niya ay lactic acid, so we have lactic acid fermentation, anaerobic fermentation, no? And that's why we have yogurt, cheese, so lahat ng mga milk products na fermented. And we also have alcoholic anaerobic fermentation, so that's why we have wine. By the way, naka-on akong ano. Okay. Again, that's why we have wine, we have beer, no, and vinegar. Ano? Ang vinegar is citric acid. Alcohol gapon. So, yun lang, no, about respiration. And try to answer questions for analysis for lesson 3. And for the next meeting, we're going to discuss translocation, absorption, and transpiration. So, again, thank you very much for joining with me. And do you have any questions or clarification before we end? Anyone? May mga tanong ba tayo or clarification? So I will just stop recording.